Hello friends, I am Shubhangi Rohatgi, PGT Bio, KV number 1 AFS Hindon, and I welcome you all to the second session of the class on human health and diseases. In the earlier class, in the earlier session, we had discussed a lot of issues related to the health and diseases. We tried to define the health, the diseases, infections, pathogens, etc. And we also studied some general characteristics of various pathogens. Also, in the later part of the session, we discussed various common diseases that are found in man. We studied about various infectious diseases caused by bacteria, fungi, viruses and other pathogens. In the last part of the session, I had given you some questions to think over and find out the answers. I hope that you have been able to find out the answers for the given questions. Now, I would like to remind you that whenever you are framing the answers for your exam, please keep in mind that your answers should be to the point and very specific. The first question that I had given you was, what are the various adaptations of pathogens that help them to survive in the host body? Please recall that we had studied the following adaptations: presence of hooks for attachment, developments to survive the pH of the gut, high reproductive potential and ability to evade the immunological responses of the host. Please try to frame your answer around these points. The second set of questions that were given to you were, what are the common modes of transmission of pathogens and how can vector-borne infections be controlled? In order to frame the answer for these questions, please recall the modes of transmission. We had studied that commonly the infectious agents are transmitted by direct physical or sexual contact, by indirect contact, by contaminated water, air and food, and by carriers or through vectors. So, in order to avoid these infections, what do we need to do? We have to simply avoid these routes so that we can prevent the diseases. The last set of questions was as follows. There were two questions. First was, how important is the maintenance of personal and social hygiene for prevention of diseases? And the second question was, our habits can save us from many diseases. Justify this statement with examples. Now, in order to frame the answer, please think that all the infectious diseases are caused by the pathogens and these pathogens propagate and are transmitted only when there is lack of personal and social hygiene. It is our habits which make us keep us healthy or unhealthy. How? Well, the simple habits like washing our hands regularly or avoiding places where there are a lot of pathogens, avoiding close contact with the infected persons or keeping the infected persons in proper isolation, avoiding close contact with them and proper disposal of waste materials. All these are the habits which we need to develop in order to prevent the infectious diseases. Please write your answer keeping these points in mind. Now, in today's session, we will discuss about the immunity and the immune system of humans. So, what is immunity? Immunity is the overall ability of host to fight diseases causing organisms and it is conferred by our immune system. There are two types of immunity, innate immunity and acquired immunity. Innate, what is the meaning of the word innate? It stands for by birth. So, this type of immunity is the one which is present by birth in the organisms. It is mainly conferred by various barriers. So, actually it is like the door which is kept shut and this door does not allow the pathogens to gain entry inside the host body. Now, the innate immunity depends on various genetic factors. The genetic makeup of the individual determines the strength of innate immunity of a person, and this is a non-specific type of defense system. It means if the gate is shut, it is shut for once and for all type of pathogens. There is no specificity present in the innate immunity. Now, let us study the innate barriers which are present. The 4 types of innate barriers are physical barriers, which include skin and the mucous coating of epithelial lining, physiological barriers like acids, saliva and tears, cellular barriers which include many different kind of cells which are in habit of killing the foreign particles present in our body, and lastly the cytokine barriers, which are certain chemicals which help the innate and acquired immune systems. Let us discuss the physical barriers in detail. So, the skin or the epithelium, it acts as the first line of defense against the invading organisms. It forms a physical barrier which is very difficult for the pathogens to overcome. This skin is impermeable to most infectious agents. Let us see how does the skin form the physical barrier and keeps the pathogen out of the body. There are many characteristics of the skin which help us to do that. First is the presence of beneficial microorganisms. In the last session also we had discussed that on our body there are many different microbes living in the microenvironment given to them by our body, and these are in symbiotic association. They might be also helpful for us. Why? Because they offer stiff competition to the other invading pathogen which is trying to gain entry into our system. Now, these microbes which are present there on the skin, they do not allow other pathogens to attack us and gain entry into our body. Nextly, the skin shows desquamation, that is the shedding. You know that the over layers of the skin are constantly replaced, and while they are being replaced, the organisms which had adhered to its surface, they get dislodged. Also, the uppermost layers of the epidermis, that is our skin, is quite dry. It does not retain moisture, and the pathogens are thus killed by desiccation. Then there are sweat glands and sebaceous glands present on the skin. Their secretions are rich in lactic acids and fatty acids. These acids make them bactericidal, that is, they kill the bacteria. Now, the linings of our tracts, that is the respiratory tract, the GI tract and the urogenital tract, they have adhesive mucus on them which can trap the microbes. This mucus also has certain antimicrobial proteins like defensins, lysozymes or surfactants which can kill the microbes, and mucus itself is quite acidic and indigestible. So, together the skin and the mucus, they prevent the microbes from gaining entry into our system. But what happens if the microbe has somehow gained entry into our system? Then it will have to encounter the physiological barriers which are present there. Physiological barriers are the chemical barriers. They are the various chemicals which are produced by different systems of the body, like antimicrobial peptides which are produced by skin and the respiratory tract, or enzymes like lysozyme or phospholipase A2 which is present in saliva, tears and breast milk, or there may be vaginal secretions which are slightly acidic, and the semen also contains defensin and zinc which are enough to kill the pathogens. Our gastric secretions are also acidic and they are also rich in proteases. All such secretions are deadly to the invading pathogens. Even if the pathogen has gained entry into the system, it will have to overcome the cellular barriers to cause a disease. These cellular barriers are various phagocytic cells that are present in the blood to kill the invading microbes or the pathogens. These cells include the macrophages, neutrophils and dendritic cells. What does the name phagocyte imply? Phago means to eat, cyte means the cells. So, the cells which are habitual of eating other things are phagocytic cells. What do? They simply engulf any foreign particle that they come across and digest it. Suppose this is a foreign particle. The phagocytic cell once locates the foreign particle, it moves towards it and engulfs it. Once the foreign particle is totally engulfed by the phagocytic cell and captured in a vacuole, the digestive enzymes will be secreted inside this vacuole, which will digest this foreign particle. For once and for all, the foreign particle will be completely broken down, it will be totally digested. Now, neutrophils and dendritic cells and macrophages, macro means big, so they are the big eaters, are all phagocytic cells. There is also one class of phagocytic cells which are polymorphonuclear neutrophils. Now, this is one long name and children do not like biology only because of presence of such names. Well, I will break it down for you. Read with me. Poly means many, morpho it refers to their structure, and with whose structure? The structure of the nucleus. So, polymorphonuclear neutrophils are those neutrophils which have many different shapes of the nucleus. This is their characteristic which has given them this name, polymorphonuclear neutrophils. And once you go into the logic of the things, then the things become very easy for you to understand and learn also. So, here is a diagram of a neutrophil and a monocyte. There are some natural killer cells also which do not attack the invading microbes directly, but instead they destroy the compromised host cells, that is the tumor cells or virus infected cells which have been invaded by the microbes. Now, they destroy these cells so that the microbes do not get to spread all over the body. After crossing the cellular barriers, the pathogens have to encounter the cytokine barriers. Cytokines are the small chemicals which induce, regulate and coordinate the innate and adaptive immune responses. The three types of cytokines are interleukins, interferons and chemokines. Interleukins are produced by the leukocytes and their function is to regulate other leukocytes, while the interferons can be made by leukocytes as well as the virally infected cells, and they contribute to the viral immunity against the viral infection. Finally, there are chemokines which direct the flow of leukocyte movement, that is the chemotactic activities. Let us discuss interferons in details. The interferons are mainly concerned with the control of viral infection. So, they can control this viral infection and viral replication inside the host cell by various activities. Some of the procedures are mentioned here. They may lead to synthesis of antiviral molecules which interfere with DNA or RNA or protein synthesis of the viruses. So, these antiviral molecules will not allow the DNA, RNA or protein synthesis of the viruses. Then the virus will not be able to propagate inside the cell. They may also produce a transitory stage of resistance against viruses in susceptible cells. and finally they may confer immunity by activating several genes that express antiviral proteins. So, these are the three methods by which interferons help us to overcome the viral pathogens which have invaded our body. This was all about the innate immunity. Let us once recall again. Innate immunity is present since birth in the individual and it is present in the form of barriers. Now, the strength of these barriers is dependent on our genetic composition, and these barriers can be physical, physiological, cellular or cytokine. These barriers try to stop the pathogen from gaining entry into the host system. But what if these barriers are overcome? These barriers are not specific also, they do not have any kind of memory. And what will happen if breaking all these barriers, the pathogen is able to gain entry into our system? Then it will have to face our second line of defense, that is the acquired immunity. Acquired immunity is the one which develops during the lifetime. Acquired means gained. So, this immunity is different from the innate immunity because it is highly pathogen specific and it also has memory. That is, once it encounters any pathogen, it will remember it all through the life and it will not forget it. And it is mainly conferred by the lymphocytes. Lymphocytes are the cells of acquired immunity. Now, let us study about acquired immunity in a little bit detail. As I told you earlier that acquired immunity is responsible for destruction of foreign particles once they have entered the body. So, innate immunity or innate immune system was there to hold the pathogens at the gate itself, to deny them the entry. But if somehow the pathogen has gained the entry into the system, then it will have to face the acquired immune system. Now, it is obviously not as good as immune system for keeping things out that it has never encountered before. It needs a little bit experience of the pathogens. Before it has seen any foreign particle, it is actually quite ignorant about how to destroy it. Once it encounters a foreign particle, then only it will learn how to overpower it. Before encountering it, it is quite ignorant what to do, how to destroy it, it does not know. So, that is why during the first exposure to an invader, the acquired immune system tries to learn to how to attack and destroy that foreign particle. But once the acquired immune system has created a response, a protective response can be made more quickly and with a greater force, allowing it to protect the body from the harm. So, when the pathogen is encountered for the very first time, the response is very slow because the immune system is getting time to know the pathogen and it is taking time to learn to destroy it. And that is why the immune response is also of very low intensity. But the next time the same pathogen enters the body and the acquired immune system encounters it, now it will produce a full on response and it will react much more quickly, so that disease cannot set in. Let us now study in detail about the immune system of the body. The immune system consists of lymphoid organs, tissues and cells, that is the lymphocytes and the antibodies. Here we are talking about the acquired immune system. Lymphoid organs are those organs where lymphocytes will be produced and tissues and cells are the various lymphocytes. Now, let us study about the cells of the acquired immunity. As I said earlier that lymphocytes are the cells of acquired immunity. They are of two types, B lymphocytes and T lymphocytes. B lymphocytes develop and mature in the bone marrow, and T lymphocytes develop in the bone marrow, but later on they move to the thymus for their maturation. That is why they are named T lymphocytes, because they mature in the thymus. I repeat once again that both types of lymphocytes are produced in the bone marrow itself, but B lymphocytes are the ones that develop and mature in the bone marrow, while the T lymphocytes move to the thymus and differentiate there, mature there. So, B lymphocytes are concerned with the humoral immunity and T lymphocytes are concerned with the cell mediated immunity. We will discuss about these two types of immunity in just a while from now. Let us first study the lymphoid organs, that is the organs where these lymphocytes are produced and differentiated and where they interact with the antigens. So, lymphoid organs, they can be of two types, primary and secondary. Primary lymphoid organs are those where immature lymphocytes differentiate. They are bone marrow and thymus. Secondary lymphoid organs are those where interaction of lymphocytes takes place with the antigens. What are antigens? Antigens are those molecules, they can be proteins or they can be sugars also, which elicit the immune response. So, secondary lymphoid organs are those lymphoid organs where the interaction of lymphocytes takes place with the antigens, and these are spleen, lymph nodes, tonsils, Peyer's patches. Let us discuss the primary lymphoid organs. As I told you earlier, the primary lymphoid organs are bone marrow and the thymus, and all lymphocytes are produced in the bone marrow, that is development of B and T, both lymphocytes takes place in the bone marrow, but maturation of only B lymphocytes occur in the bone marrow. Thymus is a structure that is located near the heart, and it is concerned with the maturation of T lymphocytes. Now, let us study the secondary lymphoid organs. The secondary lymphoid organs are lymph nodes and spleen. The lymph nodes, they actually trap the microbes and other antigens which are present in the lymph or the tissue fluid. They contain the B and the T cells and also macrophages. These are the cells which will eventually help in elimination of these antigens. Here is a picture that is given in your books also. So, you can see the network of lymphatic vessels. The lymph nodes are present all through this lymphatic system, and here the microbes are trapped which are roaming about freely in the tissue fluid or the lymph. Here they are trapped, they are filters, and now here they are presented with the B cells and T cells and macrophages which will help in their elimination. The next secondary lymphoid organ is the spleen. Spleen is the body's largest lymphatic organ, and it facilitates the immune response to blood-borne antigens. Now, see here, while the lymph nodes are concerned with the elimination of antigens which are present in the tissue fluid, spleen is concerned with removal of those antigens which are present in the blood. It also acts as a filter of the blood and removes the antigens present in the blood. It also eliminates the worn out blood cells and the platelets. Here also in the spleen there are lymphocytes and phagocytes present which will eventually remove all the antigens that are present in the blood. The next thing that we are going to study under the secondary lymphoid organs is the MALT, M-A-L-T, which stands for mucosa associated lymphoid tissue. As the name suggests, this is the lymphoid tissue which is associated with the mucosa. It makes up for about 50% of the lymphoid tissue, and it filters the tissue fluid. What will it do then? It will filter the tissue fluid, it will present them to the lymphocytes, various lymphocytes that will come there, and these lymphocytes will help in the elimination of those pathogens or antigens which were trapped there. since the name itself is telling us that it is associated with the mucus, so it is found close to the moist epithelial surfaces like the mucous membranes of the digestive, respiratory and reproductive systems. So, this was all about the secondary lymphoid organs, and as I told you, in the malt also activated B cells are present which are able to produce antibodies. Now, this brings us to the next topic, the memory of the acquired immunity. In the earlier slides also we had discussed that the acquired immunity shows the property of memory, though when it encounters the pathogen or the antigen for the first time, it takes time to recognize, to prepare the attack and to eliminate it. So, this is the primary response which is generally very slow, but whenever the same pathogen is encountered for the next time, the response will be highly intensified. This is called the secondary or anamnestic response. Now, earlier when we were talking about the types of lymphocytes, I told that B lymphocytes are concerned with the humoral immunity and T lymphocytes confer the cell mediated immunity. So, we will now discuss the humoral immunity. What is the meaning of humor? Humor stands for the bodily fluids. So, this is the type of immunity that is actually blood-borne. The humoral immune response fights pathogens that are free in the body fluids or the humors. It relies on the antigens to detect these pathogens. And what are antigens? I just told you a while ago that an antigen is a biomolecule, it can be a protein or a sugar that binds to a specific antibody. In fact, the word antigen stands for antibody generating. Antigen. So, any molecule that generates antibodies in its response will be known as antigen. Most of the times the antigens are the products of pathogens, but sometimes some antigens can also be produced by our own cells. So, the immune system should have a know-how to differentiate between the antigens which are produced by the foreign particles as well as those which are produced by our cells. We will see in later slides how the immune system prepares to do that. So, talking about the humoral immunity, the antigen leads to production of antibody. How? By the B cells. So, B cells are the major type of cells that are involved in humoral immune response. When a foreign antigen is detected, the B cells recognize the antigen and produce antibodies for fighting off the foreign invader. Now, here I would like to tell you that antibodies do not actually kill the invader, but they help in lot of ways to evade this particular antigen which they have recognized. So, up till now what we have discussed, that if there is antigen present, it will be recognized by the B cells, and now B cells will produce the antibodies, and these antibodies will recognize the antigens and help in elimination of this antigen from the system. I would like to mention here that each antibody is highly specific for the antigen for which it is produced. So, can you think how many different types of antibodies will be present in our system? Yes, many and many, because our bodies encounter a lot of antigens all through our lifetime, and as a response a lot of antibodies are produced, and each antibody is highly specific to one antigen for which it was produced. Moving on, these B cells which produce the antibodies, they cannot do it on their own, they need the help from T cells also. We will see how T cells help the B cells to make antibodies. As of now, please remember the three things, that humoral immunity is conferred by the B cells. The B cells produce antibodies in response to the antigens that they have encountered, and the antibodies are highly specific for the antigens for which they have been made by the B cells. Now, talking about the antibodies, this is the structure of antibody which is given in your book also. Each antibody is a protein molecule made up of 4 chains, 1, 2, 3, 4. So, these are the this is a Y-shaped structure. As we can see, the 2 chains are longer or larger, while other 2 chains are smaller. So, obviously, these 2 chains which are larger will be the heavier chains, and these chains which are smaller will be the lighter chains. There are many disulfide linkages present all over. So, antibody molecules is also represented as H2L2, 2 heavy, 2 light chains. Now, you can see here, while the rest of the portion, this orange portion and this red portion, this is same in almost all the antibodies, but the antigen binding sites which are shown here in blue and yellow, these sites are variable, and these are the sites which confer specificity to an antibody. Each site will be highly specific to a particular antigen that it is going to bind. Okay. So, this is the constant portion of the antibody, and this is the variable region of the antibody. Antibodies are also known as immunoglobulins and denoted by Ig, I for immuno, G for globulin, and the name globulin itself tells us that they are proteinaceous in nature, and as I have told again and again that they are highly specific. Antibodies are produced by B cells after stimulation by an antigen. That is what we had discussed earlier, that when an antigen is encountered by the B cells in response produce antibodies with the help of T cells. Now, antibodies, where are they present? They are present in the blood, in the gastric and mucous secretions, and even in the breast milk. That is why we say that they are related to the humoral immunity, the immunity which is conferred by the things which are present in the body fluids. Mostly 5 types of antibodies are known, or immunoglobulin are known: IgA, D, E, G and M. When they come in contact with an antigen, what do? They just bind to the target. Earlier also I had said that they do not directly kill off the antigen, they cannot digest the antigen, but they bind to the antigen. And what does this binding do? Actually, this binding is quite magical because it helps in elimination of antigen in many different ways. Let us see how. Firstly, it can bind to viruses or bacteria which interfere with the chemical interactions required for them to infect or bind to other cells. Blah blah blah, it is very difficult to understand. Let us try to understand it like this. Suppose this is a thief and it is trying to enter this house, and it is on its way, and meanwhile it encounters a guard dog. Now, the guard dog comes here and starts barking loudly. Will this thief be able to enter this house now? No, obviously not. Now, imagine these are the antibodies and this is the antigen. When the antibodies encounter the antigen, what do? They just go and bind to it, and now these viruses and bacteria, they will not be able to perform the chemical interactions which were required by them to gain the entry into the host cell. Now, read with me again. So, this is what I said earlier, that antibodies can bind to viruses or bacteria which interferes, this binding, it interferes with the chemical interactions required for them to infect or to bind to other cells. Similarly, in a same manner, they can also lead to clumping of various pathogens, the viruses and the bacteria that were trying to gain entry into the host cell. This was only one thing. Think about that, if there were a bunch of thieves and the dogs, instead of barking, they also corner them in one side and clump them together. Now, all of these will not be able to enter the house. Similarly, if the antibody corners the antigens and clumps them together, so the chemical interactions become impossible, and then it becomes very hard for the pathogen to gain entry into the host cell or to infect the other cells. Also, there are other methods by which antibodies can prevent the antigen for causing the harm. They opsonize the pathogen cells. What do we mean by opsonize? It means that they mark them for the destruction by the phagocytes such as macrophages and the neutrophils. To understand this, let us move back to our analogy. So, there was a thief who was trying to gain entry into this house, and a guard dog came and started barking. The dog came and it started barking on the thief, and now a policeman comes and takes this thief away. I repeat the story. There was this thief, he was trying to gain entry into the house, but a guard dog came and started barking very loudly. Hearing its bark, a policeman came and it took the thief away. So, the dog did not actually kill the thief, but it prevented the stealing of the items from the house. This is what the antibodies also do. So, antibodies opsonize the pathogen cells and corner them in one way, so that now the macrophages or other phagocytic cells come and they take away the antigen with them. Besides these, the neutralized antibody coated pathogens, they can also be filtered by the spleen to be eliminated in the urine or in the feces. Lastly, if present on the skin, they stimulate inflammation. I had told you earlier also what is inflammation. Inflammation is a type of immune response given by the body in which many things happen and which lead to swelling or redness or increase in temperature of that area or even production of pus. All these things are eventually being done in order to corner the antigen so that it cannot spread all over. These things are done to prevent the spread of the antigen all over. So, antibodies can also stimulate inflammation, and their presence in the mucus and on the skin also prevent the pathogen attack. So, these are the 5 different ways by which antibodies can act. Once again, I will repeat that they do not themselves kill the pathogen or the antigen. They are not able to do so, but they help the cellular barriers and other cells which are present in the system to get rid of those antigens. This is how antibodies work. Now, moving over to the cell mediated immunity. While the humoral immunity was conferred by B cells, the cell mediated immunity is conferred by the T cells which matured in the thymus. So, what happens that the T cells, they respond to any cell that displays markers including cells invaded by pathogens, tumor cells or transplanted cells. Any cell which shows the presence of antigens will be invaded by the T cells. I had said this earlier also that sometimes some types of antigens are also produced by the host body itself, and the T cells then identify the antigens. It does not matter to them whether they have been produced from an outside source or from the self host body, they will just try to destroy the source of the antigens. and this is the cell mediated immunity, because here the cells will be employed to kill the antigens or remove the antigens. What happens? Before going into details of cell mediated immunity, let us discuss the two types of antigens. I have been repeating one again and again that there are two types of antigens. Some can be produced by the host cells also. These cells are the virus infected cells or tumor cells or other compromised cells which have been infected by the pathogen, and now they are synthesizing the foreign proteins. Such antigens which are produced by the host cell, they are known as endogenous antigens, while the other ones are exogenous antigens. Let us see how they are formed. When a pathogen cell is digested by a phagocytic cell, this is what I had drawn earlier. When it is completely broken down, what will happen? It will broken down into small particles, and these particles, they will act as exogenous antigens. These particles which have been released from the digested pathogen. I repeat, when a pathogen is digested by a phagocytic cell, it is broken down into smaller particles, and these particles now act as exogenous antigens. The T cells are responsible for elimination of these exogenous antigens from the body. How? Let us see how cell mediated immunity works. So, first of all, the cells displaying foreign antigens bind to the T cells. Whichever cell is having the presence of foreign antigen, it will bind to the T cell, and this binding will lead to the activation of T cells. Now, further course of events will be decided by the type of the antigen, whether the antigens are exogenous or endogenous. If the antigens are produced by the digestion of foreign particle, then the cytotoxic cells will be enabled, and if the signals are arising from endogenous antigens, that is from the cells which have been compromised, which are virally infected or which are tumor cells, then T helper cells will be activated. Cytotoxic cells destroy the antigen producing cells, while the helper T cells, they lead to the production of antibodies. You remember, I had in earlier slide said that B cells required the help of T cells for the production of antibodies. This is where T cells come into action. So, if the antigen is endogenous, that is, it is produced by the viral infected cells or from the tumors or other compromised cells, then helper T cells will be produced, and these helper T cells stimulate the production of antibodies from the B cells. Antibodies are always produced by the B cells. T cells cannot produce antibodies, but they help the B cells to produce antibodies. That is why they are known as helper T cells here. And here also cytotoxic means killer of the cells. Cyto stands for cell, toxic you all know, poisonous. So, cytotoxic cells, they themselves are capable of destroying the antigen producing cells, while helper T cells, they help the B cells to produce antibodies to remove the antigen. I hope this makes the cell mediated immunity clear. Now, you must have heard that sometimes when a patient has undergone transplant of an organ or structure, he or she might have faced rejection from the body. What is this? Graft rejection. And if not about graft rejection, you must have heard about cases that whenever a transplant is needed, the donor is tried to be selected from one of the closest family members of the patient. Why? Why is it so that the transplant cannot be taken from anybody or everybody? Why only closest family members need to be the donors? Well, we know that the body can differentiate between self and non-self. How? Which type of immunity helps it to differentiate between self and non-self? Yes, it differentiates between the two with the help of antigens. The antigens that are present on the cells, they help the immune system to determine whether the particular cell is self or foreign with the help of antigens. Now, antigens are recognized by the T cells. So, the type of immunity that causes graft rejection will be conferred by the T cells, and it will be the cell mediated immunity. So, in order to decrease the chances of rejection, what can be done? Either there should be tissue matching, blood matching, and the donor should be a close relative. And yes, even after getting transplants from a close relative and even after the tissue matching and blood matching has been done, there are chances that body determines and identifies the particular organ as a foreign. If such things happen, what will occur? The immune system of the body will attack that particular organ with its full force and start killing that organ which has been transplanted, and the person may die. So, in order to prevent this situation from arising, those persons who have undergone transplant are kept on immunosuppressant drugs for a very long time. Immunosuppressant, those drugs which can suppress the function of the immune system. So, in order to prevent the rejection of the graft, the patients are kept on immunosuppressant drugs for a very long time. Now, let us discuss another type of immunity, that is the passive immunity. It is gained by the use of preformed antibodies. Active immunity, up till now we were studying about active immunity only. So, if we get to compare the active and passive immunity, in active immunity the antibodies are produced by the host body itself, and obviously it is very slow. We have been discussing that active immunity takes time to respond to a particular pathogen if it has encountered it for the first time. Now, think about a situation, if somebody has been bitten by a snake or any other poisonous insect or any other poison has entered the body, and you do not have time to wait for the body or the immune system to elicit the response. What will happen? If we keep on waiting for the immune system to elicit the response, the person might die. In such cases, it becomes imperative that preformed antibodies are administered to such patients. So, when a very quick response is required, we administer the preformed antibodies into the patient's body, and this administration of preformed antibodies from some other source is known as passive immunity. So, I have been repeating this thing again and again, that in passive immunity preformed antibodies are administered, and since the antibodies are already formed, they are in their active state. So, this response is quite faster. Whenever antidotes are made, whenever antidotes are administered, these are the examples of passive immunity. Even antibodies are present in the mother's first milk also, which is known as colostrum, and it is said that the baby, newborn baby should definitely be given that colostrum rich milk, because it is rich in preformed antibodies. The newborn baby is very delicate, the immune system is not properly developed. So, in such a condition, if a pathogen gains entry into the body, the newborn baby can become very sick. So, in order to protect the baby from the disease, the mother's milk carries the preformed antibodies, and this is an example of passive immunization. The next topic is vaccination. Immunization and vaccination, these terms go hand in hand. What is vaccination and what does it do? Vaccination is based on the principle of memory of the immune system. That is, once it encounters any pathogen, it never forgets it, and whenever it encounters the pathogen for second time, it just acts on full force to prevent the disease from setting in. The history of vaccination. Even the earliest Buddhist monks, they drank snake venom to confer immunity to snake bite. And in China, variolation was done. What is variolation? The skin tear was smeared with cowpox to confer immunity to smallpox. A small opening or wound on the skin was made, and a little amount of the cowpox virus was inserted below the skin, beneath the skin, and it was observed that the persons who were vaccinated in such a manner, they had lesser chances of getting the smallpox virus. Later, Edward Jenner, he developed the proper method. In 1796, he inoculated a 13-year-old boy with vaccinia virus, which was cowpox virus, and this boy demonstrated immunity to smallpox. Later in 1798, first smallpox vaccine was developed. So, how do vaccines work? Vaccines are basically either the antigenic proteins or attenuated pathogens. What are attenuated pathogen? Attenuated means weak. So, you can say they are the disarmed pathogens, they are the weaker strains of the pathogens which are not capable of causing the dangerous or severe form of the disease. So, what do we do? When we are vaccinating a person against a particular pathogen, either we extract the antigenic proteins or we use the attenuated pathogens and inject them in the body, and because these antigens are injected, now antibodies will be produced by the host, and when the actual exposure is there, highly intensified immune response will be generated and the disease will never get a chance to occur or set in. With the latest advancements in rDNA technology, safer and better vaccines are being produced on a large scale. Next topic is allergy. As your book reads, exaggerated response of immune system to certain antigens is known as allergy. Allergens are those molecules which are generally not harmless, and antibodies involved are IgE types, and the allergies are caused by the release of histamine and serotonin from the mast cells. The symptoms include sneezing, watery eyes, runny nose or difficulty in breathing. And for treatment, obviously since they are caused by histamines, so antihistamines, adrenalines or steroids are administered. Now, let us see what actually happens when allergy sets in. Mast cells are a type of cells which are present in the tissues that are in contact with the external environment. Those tissues which are going to interact with the external environment, they carry the mast cells. Now, IgE antibodies bind to the surface of the mast cells. If this is a mast cell, the IgE antibody will bind to its surface. Such a mast cell at which IgE antibody is attached, it is known to be sensitized. Now, whenever a particular antigen comes in contact to such an sensitized mast cell, these mast cells will cause the release of histamines and serotonins from the cells, and thus the allergy will set in. Today's modern lifestyles in which the pathogens are rarely encountered by the body, these lifestyles enhance the chances of allergy in the peoples. Last topic for today is autoimmunity. When the body's immune system fails to recognize its own cells and tissues as self, then instead of safeguarding them, it starts killing them. Immune responses are launched against these very cells. Any disease that results from such an aberrant immune response is known as the autoimmune disease, like celiac disease or rheumatoid arthritis. Why it happens is not known. Some genetic factors can be there and other causes are still unknown. With this, we have finished today's session, and let us move on to the questions for you to think over. Today we have discussed a lot of different comparisons. We discussed different types of immunities, we discussed different types of organs that confer immunities or the cells that confer immunities. A lot of comparison was made. So, I will give you just one question to do today, and that will be to differentiate between the following things. But you are required to jot down the differences, as many as you can, the differences between the primary and secondary lymphoid organs. Then we studied about the immunity. So, differentiate between innate and acquired immunity, cytokines and lysozymes only as innate barriers. Then B cells and T cells, CMI, that is the cell mediated immunity and humoral immunity, graft rejection and autoimmunity, active and passive immunity, and lastly allergy and autoimmunity. If you are able to tabulate the differences between these, then it will mean that you have understood the chapter today. Thank you.